This video will introduce the most recent research project carried out by the Wang Genomics Laboratory at the Hong Kong University of Science and Technology. The Wang Genomics Lab was founded in year 2016 by Professor Wang Wang, currently the Padma Harilelu Associate Professor of Life Science at the Division of Life Science and the Department of Chemical and Biological Engineering. In the lab, we solve biomedical problems using computation, via development and application of data science technologies and bioinformatics methods. In this particular study, our team is investigating the genetic cause of a form of brain vascular disease, named cerebral cavernous malformation. What is cerebral cavernous malformations, CCMs? The CCMs are vascular dysplasia in the brain, characterized by clusters of dilated capillaries and veins. The disease could affect as many as 0.5% of the overall population. For patients who are carrying CCMs, they have a larger chance to develop various symptoms, such as epileptic seizures, headaches, hemorrhagic stroke and other focal neurological deficits. It was widely known that about 20% of CCMs are inherited in an autosomal dominant manner, but 80% of CCMs occur sporadically, without a positive family history. These sporadic cases are termed as the simplex CCMs. Under MRI, the CCM lesions display various appearances, upon which clinicians will group patients into 4 different subtypes. Type 1 shows subacute bleeding, Type 2 exhibits popcorn-like lesions, Type 3 is featured by chronic hemorrhage, while Type 4 corresponds to dot-sized multifocal lesions. To understand the molecular mechanism of this disease, our team starts a collaboration with Beijing Tiantan Hospital. Specifically, surgeons from Beijing Tiantan Hospital resected the disease lesions of CCM patients. With the consensus of patients, we applied next-generation sequencing and droplet digital polymerase chain reaction, for example, ddPCR, to detect genetic mutations exclusively in these abnormal tissues. Overall, 113 patients enrolled in this study. Next-generation sequencing generated enormous amount of data represented by billions of short fragments of human DNA sequence. To understand the sealed book of DNA sequences, our lab developed a computation method, named MuTracer, a pipeline to identify and prioritize genetic mutations that are potentially functional in human disease. Applying MuTracer, we discovered 4 germline mutations affecting CCM1 and CCM3 and 16 non-synonymous somatic mutations, and a recurrent MAP3K3 mutation 1323C to G. Importantly, the new mutation we identified is significantly enriched in Zabramsky type 2 popcorn-like CCM lesions. The MAP3K3 gene encodes MEK3 protein, and the identified mutation leads to the replacement of an isoleucine to a methionine at the 441 amino acid position of MEK3 protein. To understand the function of this alteration, we built a 3-dimension protein structure of the MEK3 kinase domain for both the wild-type version and the mutant version. Subsequent molecular dynamic simulation in computer demonstrated the potential functional change of this protein in patients. To validate our computational prediction, we further collaborated with Peking University and Beijing Neurological Institute to perform wet lab experiments. Firstly, we demonstrated that the MAP3K3 mutation occurs in CCM endothelial cells, a single cell layer that lines all blood vessels. Then we used a widely used human endothelial cell line, named HUVEC, to confirm the downstream functional consequence of the identified mutation. Eventually, we proved that this mutation will activate the cancer-like pathway, enabling the abnormal growth of blood vessels in the patient's brain. As the kinase mutation is potentially targetable by oral drugs, it is very important to know whether the mutation occurs using non-invasive diagnostic methods. Therefore, we developed a computational model to evaluate genotype based on magnetic resonance imaging. This approach achieved high accuracy in the prediction of MAP3K3 mutation. Our discovery could help pave way for candidate drug target and therapy development, bringing benefits to patients in the near future. At present, magnetic resonance imaging (MRI) is a commonly used non-invasive method that doctors base upon for diagnosis and subtyping. A method that assesses the occurrence of potentially actionable genetic mutations from lesion appearance under MRI, providing the possibility for doctors to prescribe oral administration of medication instead of surgery, which could bear serious risks such as new cerebral hemorrhage and additional focal neurological deficits. Other projects in our lab include precision cancer medicine, machine learning, tumor microenvironment, et al. We hope to help more and more patients using the most recent AI technologies. Improving human health is the key mission of our team. If you want to know more about our research, please scan the QR code and visit our website.